Welcome back, my dear students. Today our topic is monohybrid cross. First of all, what is a monohybrid cross? Okay, it is a cross between two homozygous individuals that differ in a single characteristic. For example, you have two individuals, one is capital TT, another is small tt. Okay, this is homozygous. These two individuals are homozygous. One is tall and another one is dwarf. Okay, when these two individuals are crossed, you will get the progeny or offspring, which will be having one capital T, one small t. Okay, this cross between two individuals differing in a single characteristic, this is known as monohybrid cross. And the individual that you get after performing this cross, this is known as monohybrid, because this individual it is heterozygous with respect to a single gene, and that gene is the gene for height. Okay, now the Mendals monohybrid cross, the details of the cross which Mendel performed. Mendel started with 34 varieties of peas and spent 2 years to select the subjects, experimental plants for his experiments. Okay, he conducted selfing experiments for few successive generations to test the genetic purity of the stock plants. He carried the crosses after performing the emasculation. Emasculation means he selected two plants. In one plant, the flowers were being removed of their male productive parts, or you can say stems. Okay, this is known as emasculation to avoid self-pollination. Okay, he conducted monohybrid crosses for all the 7 characteristics chosen that we have studied earlier in the yesterday's topic. Okay, and to his surprise, he arrived at similar results in all such cases. The questions and the experimental procedure that he adopted to address these questions are here in the slide. Okay, Mendel's main question is where when pea plants with two different traits, for example round and wrinkled seeds, are crossed, will their progeny exhibit one of those traits? Will they exhibit round trait or wrinkled trait? Both of these traits, okay, are a blended intermediate trait. These were some of the questions which Mendel tried to address. And to address these questions, he adopted this experimental procedure. He used this plant as male plant and this plant as female plant, and he removed the stems from the female plant. I mean, he did emasculation just to ensure that there is no self-fertilization. Okay, then what he did, he transport the pollen grains from this flower to the stigma of that flower, and after doing this cross pollination and cross fertilization, he got the progeny seeds. Okay, these seeds were monohybrid seeds. Okay, and allowed them to germinate and raise the F1 plants. These F1 plants were then critically analyzed by Mendel, and he got the conclusions from those observations. For example, Mendel crossed a pea plant which was homozygous for round seeds with a pea plant which was homozygous for wrinkled seeds, and these two pea plants were named as parental generation. Okay, here RR, capital RR, round seeds. This is small rr, wrinkled seeds. This is known as parental generation. Okay, the offspring he got from this cross, it was possessing capital R, small r. Okay, this is known as F1 generation or first filial generation. He observed that the F1 plants expressed only one of the phenotypes present in the parental generation, and he found that the F1 plants, they expressed only the round seed trait. The plants he raised in F1 generation, they all produced the seeds which were having round seed. Mendel carried 60 such crosses to ensure that he was doing the experiments in a right way, and he obtained the similar results in all such 60 cases. Okay, he also carried reciprocal crosses and obtained similar results. That means all the F1 plants were round seeded. Reciprocal cross, I will make you understand what is reciprocal cross. For example, earlier Mendel used this plant A, this is plant B. He used plant A as male plant and plant B as female plant. Now next time he used plant B as male plant and plant A as female plant. This is known as reciprocal cross, and to his surprise he found that all the F1 plants were having round seeds. Then Mendel raised F1 generation from these seeds, and he allowed the plants to self-fertilize, producing second filial generation or F2 generation. Okay, in the F2 generation, the parental traits, I mean round seeds and wrinkled seeds, they re-emerged in F2 generation. He found these plants showed, I mean 5474 plants showed round seeds in F2 generation and 1850 plants showed wrinkled seeds. He observed that the parental phenotypes re-emerged approximately in the ratio of 3:1. Okay, that means 3/4 of the plants showed round seeds and 1/4 of the plants showed wrinkled seeds. Then he conducted such monohybrid crosses for all the 7 characteristics in P and always arrived at the same outcomes. All the F1 resembled only one of the two parents, and both the parental traits re-emerged in F2 generation in an approximate ratio of 3:1. He repeated these crosses for all the 7 characters and obtained the same results. Okay, the ratio of 3:1 in the F2 generation. This is how he performed his experiments. These were the homozygous plants possessing round seeds. This was the homozygous plant possessing wrinkled seeds. He allowed them to cross and got the F1 generation. All the F1 plants showed round seeds. He allowed these F1 plants to cross fertilize, okay, and got the F2 generation. In the F2 generation, he found that 5474 plants possessed round seeds and 1850 plants possessed wrinkled seeds. This was nearly equal to 3:4 plants possessed round seeds and 1/4 of the plants possessed wrinkled seeds. That means he got the ratio of 3:1. What was his conclusion then? The traits of the parent plants do not blend. His conclusion was that the parental traits, they do not blend. Okay, although F1 plants display the phenotype of a single parent. Okay, जो हमने देखा जो F1 plants थे वो एक ही trait दिखा रहे थे, round trait. Both traits are passed to F2 progeny in a 3:1 ratio. He found that both these traits, I mean round trait and wrinkled trait, they are passed to the F2 progeny in a ratio of 3:1. Here is the cross that you can use to better understand what he did. He crossed these two parents. One was homozygous for round seeds, one was homozygous for wrinkled seeds. Okay, this plant produced these two gametes and this plant produced the gametes. Okay, then after crossing he got the F1 generation, and F1 generation was all RR, capital R, small r. They all possessed the round seeds. Okay, then he allowed them to cross fertilize, and doing cross fertilization, they produced first of all two gametes. One is capital R, then small r. Okay, here also capital R and small r. Then you can see in the F2 generation he obtained plants in the ratio of 3:1. These were the plants that possessed round seeds. Okay, because they possessed this capital R. Okay, which controls the formation of round seeds. And this percentage of individuals, they did not possess any capital R, they were having wrinkled seeds. Okay, so you can simply say that जो monohybrid cross किया Mendel ने, he arrived at a conclusion that the traits, okay, the parental traits, they are passed to F1 generation. Okay, and the F1 generation expresses only one of these two parental traits. Then these traits they remerge in the F2 generation in the ratio of 3:1. This is all about monohybrid cross. I hope you all understand this. See you soon next time with a new lesson. Till then, God bless you and thank you.